So I got this beautiful picture of flower. I'm not sure what it is, but one of my favorite things, flowers. I just got some last night, put over in the house. Anyway, in the flower, we have these nail parts here holding the pollen, and you can't quite see, but the ovary would be over here, the stain, the steel and ovary. And what does it have to do with gametes? Well, remember, gametes in humans are sperm and eggs. There's the reproductive cells that the sexually producing organisms need to make. And here you can see all the pollen. They're haploid. Gametes for these flowers are being produced. Hopefully these will get rubbed off. Other insects can then spread them around other flowers or fly off on the wind. Either way you go, flowers want to take this pollen and give it to another flower, so you can reproduce sexually. So these are gametes. You're looking right at some pollen right here that are haploid, that are ready to go fertilize. Here, I believe this is, which Kardashian? Not, it's Kourtney. Khloe. I think it's Kourtney. Get it? I spell it right? Anyway, genotype, difference in genotype and phenotype. If you look at Kourtney Kardashian, you can see a widow's peak, which means she's either capital W, or capital W, little w. We don't know from looking at her, but we know her phenotype. So that's the phenotype, widow's peak. But the genotype, we don't know. We don't know if she's this or this. So the genotype are the actual genes. Phenotype is what they look like, the appearance, which I think I have it drawn below. I went ahead and put the whole, I did some serious research on the Kardashian images, the two parents of the three, although I think Khloe, this is Khloe, right? We all know that's Kim. This is Kris. Oh my gosh, the gaze, that's kind of a little weird. But anyway, no judgment. And Kourtney. Okay. Oh, I messed that up. I covered exactly what I was supposed to write there. Oh God. So, what I was going to say, no, I messed up. I've accidentally progressed it. So I'll have to see later. But if you look at the two parents, they have a widow's peak. You notice that. So both parents are either, this, or one of them is homozygous dominant for widow's peak. If both parents are heterozygous, we can do a little Punnett square to figure out how many kids should have a widow's peak. And if we look at this, 75% of their kids should have a widow's peak. And if you look, Kourtney definitely does. I can't really tell about Khloe, but I think Kim, I suspect Kim naturally has a widow's peak. It's very hard for me to find it. I think if you go back and find images of her before she started hanging out with Paris Hilton, got famous, you find Kim Kardashian with a widow's peak. That's your assignment. So Kim Kardashian, widow's peak. Oh, you can't read that, but you heard me. Find that picture. I think it's out there, because I would not put it past Kim Kardashian to remove that, have it remove the hair follicles, because you know how obsessed she is with her appearance. We're talking about her, or I am right here. Anyway, so we talked about hemophilia, and we talked about recovering the Romanov. So here's just a diagram showing what happens. So you have injury of blood vessel, you will bleed. Okay, So naturally your vessels will constrict, and then you have these clotting factors activating your blood from that oxygen coming in. Say, hey, we got a problem here. And with the clotting factors and platelets, you get a strong fibrin plug to form. Yes. Yummy. All right. You have that fibrin. Normally, you got the fibrin clot. Look at that. Oh, clotting that up. Boom. Stop that blood. Stable blood clot. Or, I'm sorry. Fibrin. Stable fibrin clot. Fibrin. That's the stuff right here. And if you have hemophilia A, a weak platelet plug. See, that's a little weaker. You don't have as many platelets going in there, and then a weaker plug. So, more likely to lose blood, and that's a problem. So this is what used to happen before DNA testing as far as paternity. You got, a child, you want to know who the father is. I mean, who wouldn't? So, you have up in here, we have the mother, we have the father and the mother here. And if you have A-blooded parents, you can have, of course, A-blooded child, or, if they're both heterozygous, those O's can come together, you have O-blooded kids. Over here, 2 O-blooded parents, you're only going to have O-blooded kids. Here is interesting. So, if the mother's, I'll get them. The mother's blood is B, and the father's blood, if they're both B, it's pretty interesting. The father could have A blood because he could have that O allele, and that could combine. He could be B blood, right, where he contributes that B allele. He could be AB, contributing that B allele, or he could be O blood, because after all, the child. So this would not help you if before you had DNA, the mother was B and the child was B. There's no way you could do a blood test to see if even eliminate. So this blood test for paternity, could eliminate but not prove paternity. And over here, if the child was O blood, he could be A, contributing O, or B, contributing O, or O blood, of course, but not AB, because AB, that A or B will get through, and we get through to the offspring. Now we have this is a map. You're seeing the percent of the B allele you see in the world, and you see North and South America, not very many, very low percentage. That B allele is really isolated to this part of Asia, China, India, and this part Central Asia. Most of this is Russia right now. And these hills here, oh yeah, India, look at that. Nepal, this is where Mount Everest is. So it kind of varies. It doesn't mean you don't find a lot of people with B allele here too, because the population. Here's hemophilia. Remember, you show this, on the X chromosome, the big H is normal, little h is hemophilia. And, it's kind of fun, not fun for them, but fun for us to kind of look through the royal family. Look, everybody who has hemophilia, guess what? What do you notice? You're saying to yourself, they're males, okay? And you have them carrier females here. If you're colorblind, you will not see anything here. You'll just see a bunch of dots. But if you're not colorblind, you can trace the outline of a nice little boat right there. Here's some good, cool shots. Most male, this is the most common here. Most common. I have 2 good friends. Best man at my wedding and best friend in California when I moved here. Both of them red, green colorblind. This is how they would see colors. So you see yellows, blues pretty well, no problem. Just the reds, the pinks, oranges are a little problematic for them. But you would never know unless they told you. And this is what a yellow-blue colorblind is. And here's also kind of cool. Shows you if they looked at it. And make sure you can do Punnett squares like this, the way I filled it out. You see, bring the X's down and bring, this is for sex-linked traits. This is color blindness, and this is hemophilia. And we did it several times with the covering the Romanovs. You can see how this male has hemophilia, this normal female, not a carrier, no problem. No problem with the kids, but all of the females, because they all got their X from their father, and that father gave them a hemophilia allele on their x-axis. And for sex-linked, similar thing. Well, here the mother's on top, and she's carrier, normal father. Oh, it's kind of counterintuitive. They have a male. Where did the male get his colorblindness from? From the mother, not the father. So those are things to be counterintuitive. So make sure you just work out a Punnett square for this stuff, okay? And you write those out. And then they might ask you about genotypic ratios and phenotypic ratios. Know what the difference is. Genotypic of the genes. How many different genotypes do you see? Here's X's with two big B's, X's with big and a small. X with a big B, X with a little b, and a Y. So there's 4 different ones. That's a 1 to 1 to 1 to 1. But the phenotypes, there's only one colorblind out of 3. So that's a 3 to 1 ratio. And here you got 2 that are the same. These are identical. These are identical. So that's a 1 to 1. And phenotype ratio, look, these are 2 carriers and these are normal.